Hello, everyone. Uh, First of all, I'd like to thank Kathleen and the other organizers of this nice online aquaporin workshop for giving me the chance to tell you about our most recent work on the role of aquaporins in environmental and hormonal signaling in particular, as they serve as both targets and players. And this is typically our current research focus. So over the years, my team has been trying to understand the role of cell membranes and more broadly tissue hydraulics in maintaining the plant water status. And for this, we have been dissecting elementary transport processes, for instance, in root, trying to understand how water flows from the salt solution into the xylem vessels. We observe a very similar process occurring in leaf veins when water flows out of the vessels to cross several cell layers before reaching the stomatal chamber. And also, we can anticipate very important role of cell membranes during change in gut cell volume and stomatal movements. So over the years, we have been considering different types of stimuli were able to change the hydraulic properties of all these systems. And here you see a list of all the signals and stimuli that we've been studying. And our aim was really to understand what might be the role of aquaporins and in particular, a member of the PEEP subfamily in these regulations. So today I'd like to start first by showing you measurements done by Karin Prado, a very talented graduate student in my group, who characterized the hydraulic conductivity of the Arabidopsis rosette using pressure chamber measurements. So Karin was very patient to perform these measurements over time in day and night cycles. What you see in these conditions is a slight oscillation of the rosette hydraulic conductivity, so-called KROS, over time. However, we kind of discovered by accident that if we maintain the plants in darkness after a night, so this is shown here, what we observe is that we have even more pronounced oscillations with a peak that would correspond to subjective midday. And in these conditions, we can reveal a true circadian oscillation of the rosette hydraulic conductivity with a peak at subjective midday. So of course, we're very keen on understanding what might be the molecular basis of these oscillations. And of course, we have characterized which kind of aquaporin isoforms, in particular, which PEEPs could be involved in this response. And here you see the name of the three PEEP isoforms, which somehow play a role in KROS regulation. And as you can see, they share common expression in the leaf veins we have shown are very important for this response. So this is an illustration of the type of measurements that we did in a knockout mutant for PIP21, showing how this mutant is altered in the response to light and darkness with a peak in KROS at subjective midday and the darkness. So this response is completely missing in the PIP21 mutant, indicating a very important role of this aquaporin in this process. So further on, we wanted to try to understand what kind of regulation could account for these oscillations. And we've been considering the role of P21 abundance and phosphorylation. And for that, one of the approaches, in addition to proteomic approaches, was to raise antibodies that are recognizing either the complete set of P2 proteins, so the so-called anti-P2 antibodies, but we also rose antibody directed against this C-terminus, which recognize either the C-terminus as unphosphorylated form, so anti-0P, or in the dephosphorylated form, anti-2P. So here in these immunodetection studies, which correspond to different time points, we could realize that in all conditions, the level of the P2 aquaporins is pretty stable. However, if we consider the ratio between the 2P forms and the 0P form, we see a very marked oscillation and peaks at subjective midday, indicating that the rise in aquaporin phosphorylation as these two sites is quite nicely in phase with the oscillation of KROS. So this really supports the role of phosphorylation in mediating this change in aquaporin activity, which in turn would reflect and use this change in KROS. But we wanted to have more precise genetic evidence for the role of aquaporin phosphorylation in these processes. And this is the reason why we have designed mutation forms of PIP21, either phosphoformic form, where the two serine residues were mutated to aspartic acid, and phosphodeficient forms, where we have introduced serine to alanine mutations. And these two forms were reintroduced in the PIP21 background. So here you see the typical response of core zero plant to subjective midday increase in KROS in these conditions, which is missing in the PIP21 mutant. Reintroducing the wild-type protein restores the response. However, if we reintroduce the phosphodeficient form, we lack the stimulation of KROS under darkness at subjective midday. In contrast, we can restore this response in the plants expressing the DD form. However, you could realize that this phosphomimic form is somehow necessary to get the response and the increase in KROS, but not sufficient because it's we still see a low KROS in these conditions. So indicating that there is probably another component which would mediate the activation of the aquaporin in this system. And to make a long story short, I can tell you that we have been searching for partners interacting proteins which might be able to interact with the C-terminus of the P2 aquaporins to possibly activate. And we worked as candidates with 1450 proteins and could show that indeed these proteins are able to interact with this aquaporin in the phosphorylated status to activate them. So then we are come up with this kind of model where we see a typical oscillation and change in the aquaporin level, but in addition to have a full activation of the aquaporin at midday, we need the additional role of 1450 protein, which by molecular interaction will further activate the aquaporin. So now I'd like to move to studies that we did in roots. Although we did a lot of such studies today, I will just show you one slide related to work performed by Zagam Shazad, where he studied the effects of ABA on root hydraulic conductivity that again we measure using pressure chamber techniques. So here you see that a treatment of roots and plants by 1 micromolar ABA for 3 hours induces a significant decrease in root hydraulic conductivity, so-called LPR. So why being interested in this response? Well, there was some information from literature that these effects of ABA could be mediated by changes in aquaporin phosphorylation. And we use the same tools as I showed before, using these phosphospecific antibody rates against the C-terminus. And in these conditions, what we see is that the antibody that recognizes the non-phosphorylated form shows an enhanced signal after ABA treatment, whereas the antibody that recognizes the dephosphorylated form shows a decreased signal, indicating that this decrease in root hydraulic conductivity as induced by ABA is related to a decreased phosphorylation of the P2 C-terminus. So this is another example where an hormone is able to act on aquaporin activity through phosphorylation. Well, we found another context where ABA seems to be very important for aquaporin function, and this comes from studies where we investigated the role of aquaporins in stomatal movement. So our approach was to screen different types of aquaporin mutants using this pill epidermal stomatal assay, and we found that plants lacking P2-1 have a typical response, opening response to light, low CO2, they close properly in response to darkness or high CO2, but they kind of fail to respond to ABA. And this is shown here. You see a typical opening response of stomata in response to light, and here a typical closing response to ABA as shown in the wild-type plants. But these two allelic mutants for P2-1 show a very marked defect in the closing response. So this led us to, in this system again, investigate in closer details the effects of ABA on water transport. And for that, we use a protoplast assay where we isolate guard cell protoplasts and we measure the osmotic water permeability using this micropipette. So here you see typical swelling curves of protoplasts in response to a hypotonic treatment. And from the rate of this swelling kinetics, we can deduce the osmotic water permeability of the protoplast, which, as you can see here, is strongly stimulated by ABA treatment of the protoplast after 1 hour. So in contrast, when we perform these experiments in the two allelic mutant lacking P2-1, these protoplasts typically lack this enhancement in PF, indicating that ABA mediates this ABA-dependent increase in guard cell water permeability. So later on, we tested the idea that maybe this kind of activation role of P2-1 could be linked very closely to the ABA signaling system. And we investigated the capacity of ST1, which is a kinase which plays a very crucial role in ABA signaling. We tested the capacity of this kinase to directly phosphorylate the aquaporin. For that, we use in vitro phosphorylation assays with different peptides representing this putative site and, of course, the C-terminus of the protein. Here you can see that the C-terminus was clearly not recognized by the OST1 protein kinase. In contrast, the OST1 was able to phosphorylate this loop, intracellular loop, so-called loop B, and this phosphorylation response was missing when we mutated this serine 121. So indicating the functional role, putative functional role for another site in the aquaporin. So we tested different approaches to validate the functionality of this site, and one of these approach was, as I mentioned before, to introduce phosphodeficient and phosphomimic forms of this residue. So, and again, these different forms were introduced in the PIP21 background. So here again, I recapitulate the lack of response in protoplasm to ABA in the mutant, which is this response is enhanced if we overexpress PIP21. And by introducing this phosphodeficient form, we cannot restore the ABA response. In contrast, when we introduce phosphomimic forms with different expression levels, we see a constitutive increase in the water permeability of the guard cell protoplasm, indicating that in this case, the phosphorylation of this residue is necessary and sufficient to increase the osmotic water permeability of the guard cell plasma membrane. So you might have been surprised of this very dramatic lack of closure response in the PIP21 mutant. And for us, that was quite a puzzling question to realize that a simple defect in hydraulic and water transport could result in such a slowdown of the closing response. So for these reasons, we have considered another possibility that PIP21 could play another type of role in the guard cell response, possibly involving signaling itself. So we know that the PIP aquaporins are possibly permeable to H2O2, and we investigated this function. So we know that in response to ABA, there is a production of reactive oxygen species, which in turn will accumulate in the guard cell. And we follow this response by using the DCFDA dye, and you see this typical accumulation in the guard cell, which is missing in the 2 allelic mutant for PIP21. So in addition to these experiments, we are using this biosensor, this protein, which is more specific to H2O2, and we got a very similar response indicating that in the core0 background, we see this typical accumulation of H2O2 in the guard cell in response to ABA, which is missing in the 2 allelic mutant, supporting the idea that indeed PIP21 plays a true role in H2O2 transport. So in conclusion, these studies in guard cells have revealed a dual function of PIP21 in water and H2O2 transport. And here we have found that in response to ABA, we get a very efficient activation of OST1, which in turn will activate PIP2 one aquaporin on the plasma membrane. We know that OST1 will also activate the NADPH oxidase, which ultimately will lead to the accumulation of H2O2 in the apoplasm, which will then be flowing through the aquaporin. This is the first signaling function that we propose for the aquaporin. But also, the aquaporin could also contribute to the water efflux. So I didn't have time to further mention, but what we have observed is that the fledgling 22 elicitor play a very similar role in this scheme, and through OST1 activation is able to activate a very similar pathway, indicating that PIP21 play a very general role in guard cell signaling. And in the recent year, that was for us the very most exciting result that we got in the aquaporin field. So another important message of my talk was to find that and to show that the single aquaporin isoform can show very specific phosphorylation response depending on the organ and the stimulus. I showed you the very critical roles of this serine 121 site and the role that the C-terminus phosphorylation plays in different contexts. So now a very unsolved, important mystery for us is to understand what kind of upstream events are able to make this distinction between these type of regulations, and on the long term, how response in the root, in the leaf veins, and in the guard cells can be linked to have an integrated response to water deficit in particular. So I'd like to close this talk again by thanking my collaborators. So I mentioned different names. So here you see different members of my group. And I'd like also to thank our collaborator, Nathalie Leonard, for studies and on guard cells, as well as Yusuke Saicho, and Tony Scheffner is a long-term collaborator for providing different types of materials. And I thank you for your attention.